Hello, I'm Dr. Greg Ketchum, principal of Talent Planet, a leadership development and media production firm. And I'm joined today by Dr. Tony Frudakis, who is the Chief Scientific Officer of GLG Pharma. Welcome, Dr. Frudakis.
Yeah, thank you very much.
May I call you Tony?
Absolutely.
All right, Tony. So I understand you and your colleagues are working on a new and very promising targeted form of cancer therapy. Can you tell us a little bit about that?
Sure. Well, as you probably know, cancer is an insidious disease. We've been battling it for many, many decades within the context of recent scientific developments and made very little progress, disappointingly. The first generation of oncology treatments were based on killing all dividing cells indiscriminately, the chemotherapeutic treatments. These are associated with very low response rates, typically about 25%, 4 to 6 month duration of response, 6 to 9 month survival benefit, very meager. With the genetic revolution, we've advanced to more of a precision oncology approach, and there's a lot of excitement about that because cancer is a very heterogeneous disease, meaning that the genetic architecture or the combination of mutations, over and under expressed genes, which genes are translated, how they're modified, all of that put together is different from each patient's disease to the next. So even two patients with breast cancer could have almost two completely different diseases based on their genetic architecture. And so the one pill fits all approach of developing and approving therapeutic compounds or drugs based on how large populations of patients respond is inherently defective and explains partly why we have low response rates. But with the precision oncology approach, the idea is to consider the patient's genetic architecture when deciding which treatments would be best for that patient. What's going to be needed eventually, and this is where GLG comes in, is a complete personalization of that process. We need to consider the patient's disease as a unique disease, just as unique as their physical appearance, their personality. It's a unique thing. And the way to do that is to actually take the patient's disease out of the body and study it, to culture it, to test it, to see which drugs work best for that tumor or cancer and which do not. And there's been recently progress in this area. It's called an ex vivo approach, where the cancer cells are transplanted into a mouse and grown. And this is the general idea, but the problem with that approach is that it takes so long and it's very expensive, and the tumor cells are in a foreign environment inside of the mouse, and they eventually lose their identity.
Okay. It sounds pretty comprehensive. It sounds like, though, the main difference in what's come before and what you're doing now is this is a very, very personalized approach to the individual person, as opposed to a population of people who have a disease that has the same name. Is that correct?
Yes, exactly. That's the key, is to consider the disease as a unique entity when deciding on therapeutic options.
Are you all taking a slightly different approach than what's been done before with this kind of targeted therapy?
Yeah, so as I mentioned, there's been some recent developments by transplanting patient diseases into mice so that they could be studied specifically. This is an empirical approach. Okay. Let's see what works with your cancer, rather than let's just make assumptions based on which genes or a few gene sets are being expressed or not expressed. The problem with the empirical approach, as I mentioned, is that it doesn't quite get the job done yet, because the only way to keep these cells alive outside of the patient's body is to put them into a mouse, which is a foreign environment for that patient's tumor cells. And so they change very quickly. And all of the results that are learned by studying those cells become quickly irrelevant to the patient. What we at GLG have done, along with our partners, is developed a platform which potentially solves that problem. We take patient cells out of the body through resection or biopsy, and we can culture them outside of a mouse in a three-dimensional microenvironment. We actually can identify all of the biomimetic, they're called, mimicking life, biomimetic peptides that patient's cancer was reliant on. We can mass produce them, and we can incubate them with the patient's cells in a three-dimensional nanoporous microenvironment to recreate as more perfectly than has ever been done before the natural tumor microenvironment from which the cells were taken. So this is key. Now we can do all sorts of things. We can spread the cells out onto large arrays and test them with very large numbers of drugs and drug combinations. And in fact, what we want to do is test those cells against our very specific compound libraries, which are based around the STAT3 protein, inhibition of the STAT3 protein. STAT3 is one of the key genes in the human genome that's used for normal embryonic development, but it also is misregulated in a vast majority of cancers. It's considered a cellular gateway to cancer. And so our approach is to take a very special collection of STAT3 inhibitors, which are unlike any other inhibitors ever described because they're very specific to the STAT3 protein and their toxicity is low and well known, to take those and combine them with a large number of other what are called pathway-targeted therapeutics and general chemotherapeutics to find the quote-unquote magic cocktail for each patient.
Okay, that sounds really fascinating and pretty complex to me. Now, in my experience, people usually get involved with worthy causes like this because of some personal meaning. What's the meaning for you in this work?
Well, it's frustrating to know that we have the ability to do so much better than we actually do, and it's not just having had friends that were dying and knew that I had a background in cancer research and asking me what they could do, and me feeling impotent because I couldn't help them because there were no clinical trials designed in the way that I'm talking to you about today, and most of the trials that they had access to had the results speak for themselves. Even the best trial results are about 50%, so it's still a 50-50 shot with with most of the patients not enduring indefinitely. So there's still a possibility of relapse. So in any case, it's not just that I couldn't help particular friends. It's just more of a general frustration that we could be doing so much better. It's sort of like having a computer and having an abacus being asked to solve a math problem and not being able to use a computer. Mm-hmm. Very, very frustrating.
I understand that. Well, how can our viewers help? How can they get involved?
I think that if people were to continue to voice their desire to see things improve, to see scientists, companies, academic institutions, and regulatory agencies all working together to promote the pushing of the envelope. And we would seem to have little to lose. We have all this great technology. Let's throw it at the problem. Mm-hmm. Let's see what we can do. And so, this is why I'm excited to work with companies like GLGs, because we're literally trying to force a transformative change in a very staid, conservative marketplace. That to me is exciting. That's where the attention is needed, is in these particular markets where change is very slow to come.
Understood. So if our viewers want more information, Tony, where would they go to find that?
glgpharma.com. That's our website. That would be the place to start. We have an active social media campaign where we talk about recent developments and other approaches that are what we believe approaching the problem from the right angle, the empirical angle rather than the reductionist angle. So it's sort of food for thought. There's opportunity to opine and to be educated, and a lot of fun for the layperson and the scientifically inclined alike.
Absolutely. Well, as a cancer survivor myself, I really appreciate the work that you and your colleagues are doing, Tony. I've been talking with Dr. Tony Frudakis, who is the Chief Scientific Officer at GLG Pharma. Tony, thank you so much for being with us today.
Thank you very much.